Conventional trials to study the efficacy of various protocols require that human subjects be intentionally exposed to the pathogen under investigation. Now, obviously, that convention is not feasible in the case of Ebola virus disease. And so for that reason, in the United States, the Food and Drug Administration, the FDA, has established the Animal Efficacy Rule, or simply Animal Rule, which allows for limited approval for medical research using animal models to replicate human disease. And by the way, the Animal Efficacy Rule was authorized by the United States Congress in 2002 in response to the terror attacks of September 11, 2001. So with that in mind, let us consider the following EVD treatments: ZMapp, favipiravir, BCX4430, JK05, and MIL77. ZMapp, named for MAP Biopharmaceuticals, one of the developers of the drug, is a cocktail of 3 antibodies that act to neutralize Ebola virus proteins, thereby conferring passive immunity to the patient. The antibodies were first produced and subsequently harvested from mice. The antibodies were then transfected into tobacco plants for replication. While the antibodies have been used successfully to treat Ebola virus-infected monkeys, ZMapp was used to treat a small number of human patients during the 2014 event. However, the results of those treatments were less than stellar. But after further modification, ZMapp is currently being tested in a randomized controlled trial in Liberia and the United States. And that study, by the way, was begun in February 2015. So here are some important points to remember about ZMapp. ZMapp consists of 3 antibodies. The antibodies neutralize Ebola virus proteins. It has been used to successfully treat infected monkeys, and it is currently being used in a randomized human trial that was begun in 2015. Favipiravir. Favipiravir is an experimental antiviral drug developed in Japan. It is effective against many RNA viruses, including influenza, West Nile, yellow fever, foot and mouth disease, and even against the Rift Valley fever. Favipiravir's mechanism of action is the selective inhibition of viral RNA-dependent RNA polymerase. Favipiravir does not inhibit RNA or DNA synthesis in mammalian cells, and therefore it is not toxic to humans. Now in 2014, Japan began stockpiling favipiravir against future influenza pandemics. Favipiravir has been shown effective against Ebola virus in mice, though its effectiveness in humans is not yet proven. Preliminary results from a 2014 clinical trial in Guinea showed decrease in mortality rate in patients with low to moderate levels of Ebola virus in their blood. So, important points to remember about favipiravir. It was developed by the Japanese. It is effective against numerous RNA viruses. It has been used successfully to treat infected mice, and the results of a preliminary human clinical trial started in 2014 are very promising. BCX4430, also known as Imucillin A. Imucillin A is a broad-spectrum antiviral drug developed with funding from the National Institute of Allergies and Infectious Diseases, NIAID. Originally intended as a treatment for hepatitis C, another viral infection, Imucillin A was subsequently developed as a potential treatment for deadly filovirus infections as a broad-spectrum antiviral. Broad-spectrum, that means it is effective against a variety of pathogens. Imucillin A is effective against a number of other RNA virus families. Imucillin A has been shown to protect against both Ebola virus and Marburg virus infections in both rats and monkeys. And here's a neat thing, even when it was administered up to 48 hours after infection. Imucillin A's development for use in humans is being fast-tracked in light of the 2014-2015 EVD outbreak. Here are some important points to remember about Imucillin A. It is a broad-spectrum antiviral. Remember, broad-spectrum means it's effective against many different viruses. It was originally developed as a prophylactic for hepatitis C. It is effective against Ebola virus and Marburg virus in rats and monkeys, and remember, even after 48 hours, and human trials are still pending. JK05. JK05 is a broad-spectrum antiviral drug developed in China this time in conjunction with the Chinese Academy of Military Medical Sciences. Now, JK05 is an inhibitor of the viral enzyme RNA polymerase, which you'll remember, I'm sure, is essential for viral replication. And remember that in the Ebola virus genome, it's the L gene that codes for RNase. JK05 has been tested in mice and has proven effective against several RNA viruses, including influenza, Ebola virus, and yellow fever. Chinese authorities now have not published the chemical structure of JK05, but it is reported to be suspiciously similar to that of Japan's favipiravir. In fact, WHO officials believe JK05 to be an exact duplicate based on the favipiravir patent. At any rate, JK05 is a small molecule with a simple structure, and those qualities lend themselves nicely to synthesis scale-up for mass production in short order. Now, JK05 has been approved by Chinese authorities to be available for Chinese healthcare workers involved in combating the 2014 EVD outbreak. So, important points to remember about JK05. It's developed in China. It's a broad-spectrum antiviral. It is very similar to favipiravir, and it also inhibits viral RNase activity. MIL-77. Now, MIL-77, not unlike its North American cousin ZMapp, is an experimental cocktail of antiviral antibodies that act to neutralize Ebola virus proteins, again, like RNA polymerase, effectively halting production of virus structural components within host cells. Now, MIL-77 was tested in 18 monkeys infected with Ebola virus, and according to released results, all of the monkeys survived. Now, you should recall that MIL-77 was first used to successfully treat British military nurse Anna Cross in March 2015. We talked about her some time ago. And finally, important points to remember about MIL-77. It is similar to ZMapp. It is a cocktail of antiviral antibodies. It neutralizes viral RNA polymerase, and it was used for the first time in Great Britain in 2015. Now, a number of vaccines against Ebola virus were already in development prior to the 2014-2015 outbreak. Recall that vaccines initiate the development of antibodies to specific antigens by the immune system. Several of those vaccine candidates had been shown to protect non-human primates against lethal Ebola virus infection. None, however, had been approved for clinical use in humans. As of early 2016, there were at least 13 different EBOV vaccines in various stages of development or testing. I want to look at the top 3 candidates now. So in September 2014, clinical trials began for the CAD3 EBOZ vaccine. Now, the EBOZ here denotes that the vaccine is specific for Zaire Ebola virus. CAD3 is an attenuated version of a chimpanzee adenovirus. That's the CAD3 part. Attenuated simply means that the virus has been genetically altered so that it is unable to replicate in humans. Now, in one trial, 20 volunteers from Bethesda, Maryland, USA, were injected with the attenuated virus, and they were appropriately monitored. In November 2014, initial trial results were released, and those were in fact quite promising, as all 20 participants had developed antibodies to the Ebola virus surface glycoprotein antigen. Then in October 2014, multiple trials of the experimental VSV EBOV vaccine commenced in Europe, Gabon, Kenya, and the USA. This vaccine consists of the attenuated human vesicular stomatitis virus, that's the VSV part, which was modified to express again a surface glycoprotein of Zaire Ebola virus. In April 2015, a Phase 3 trial of VSV was begun in Liberia following a successful Phase 2 study in that country. Now, the College of Medicine and Allied Health Sciences in Sierra Leone, the University of Sierra Leone, the Sierra Leone Ministry of Health and Sanitation, and the Centers for Disease Control and Prevention here in the United States together launched STRIVE in April 2015. STRIVE is an acronym, and it means the Sierra Leone Trial to Introduce a Vaccine Against Ebola. It's designed to assess the safety and efficacy of a candidate Ebola vaccine, that's the VSV, among healthcare and other frontline workers. Preliminary results of a Phase 3 study in Guinea released in July 2015 indicated that the VSV vaccine appeared to be safe for general implementation, and the VSV vaccine is now being used in West Africa routinely as the trial continues. And finally, Johnson & Johnson here in the United States, in conjunction with its Denmark sister company Bavarian Nordic, has developed the AD26 ZEBOV/MVA-BN vaccine. I will break that down for you. Now, this Ebola virus vaccine consists of two parts, the prime and the boost. Now, the prime component is derived from human adenovirus serotype 26, that's the AD26 part, which expresses a Zaire Ebola virus surface glycoprotein, while the boost or booster component is a modified Vaccinia Ankara virus developed by Bavarian Nordic. Modified Vaccinia Ankara, Bavarian Nordic, that's the MVA-BN part. In January 2015, preliminary Phase 1 trials indicated that participants developed temporary immunity in accordance with expected results of the vaccine's prime component, so it worked. And Phase 2 trials were initiated in the UK, France, and Sierra Leone in late summer, early fall 2015, and those trials remain ongoing as of now, early spring 2016. In our next lecture, we will consider Ebola virus in light of weapons of mass destruction, biological warfare, and bioterrorism. I'll see you in the next lecture.